Muhammad Umar Farooq biological video. Please make sure to subscribe, like and share. Ask the question for answer and comment about the artwork. You can also answer question to each other for solving the problem in the comment below. So here today we will discuss about the control of gene expression in eukaryotes. So the eukaryote is different the control of gene expression. We will just understand the eukaryote which control the gene expression. So the expression of gene mean the DNA will express the genotype expressed in the form of phenotype to show their functional properties. So the control of gene expression in eukaryotes is made up of four points. Here we will discuss the transcriptional control of gene expression and post-transcriptional control of gene expression. On the other hand, more is the translational and post-translational control and role of hormones in control of gene expression. So the post-translational control of gene expression we will discuss, and translational and post-translational control and role of hormones in control of gene expression. We will discuss just overview. So here is the transcriptional control of gene expression. First of all, we will discuss. So the general concept about the control of transcription to translation is here. Here is the nucleus and here is the cytoplasm. So the nucleus contains chromosome in the form of DNA, and the DNA will transcript into RNA, and this pre-RNA convert into the messenger RNA after the splicing to release the intron, and exon will be left behind. So that go into the cytoplasm and bind with the ribosome and transfer RNA to synthesis of protein that is in the form of polypeptide. So the many ribosomes are attached is known as the polysome. So here is the messenger RNA which that is showing in the blue color. While here we will discuss that is the transcriptional control of gene expression. So the RNA polymerase and transcriptional factor that bind to enhancer and transcriptional factor that bind to RNA polymerase, which that is initiation factor, which that initiate the gene expression in the messenger RNA, that is the transcription initiation. So let's talk about here is the DNA. DNA is made up of polynucleotide. So this polynucleotide double-stranded RNA is looking like that. So this promoter region which that is bind this RNA polymerase and transcription factor that bind to RNA polymerase. So the transcription factor that bind to enhancer. So here is the enhancer sequence which that is used for the binding of transcriptional factor that bind to enhancer for enhance. So here is the promoter and coding region for gene. So that is all thing is in the DNA in the present in the nucleus. So that nucleus contain this DNA which that produce RNA, that is known as the transcription. So here is the DNA which that is present in the nucleus, and this transcription factor will bind to trigger the transcriptional factor and RNA polymerase bind. So that's an enhancer sequence which that stimulate the transcription to begin. So this transcription in the form of RNA, that's RNA is the pre-RNA. Pre-RNA means the exon and intron are present in the RNA, while that is modification by the splicing enzyme. So this splicing enzyme used for it. So let's talk about here is the nucleus we will discuss. This is the post-transcriptional control of gene expression. So the after the transcriptional factor, what will be the necessary things for the controlling to the complete messenger RNA? So that is the post-transcriptional. Post mean after and pre mean before. So here is the post-transcriptional control of gene expression we will discuss. So that here is the nucleolus, which that is the part of the nucleus, while here is the chromosome are present in the nucleus, which that is made up of DNA. So here is the DNA thread I am taking out. So the transcription, that is the transcription to produce the RNA. This RNA is two-sided, 5' and poly A tail. So poly A tail mean the adenine, tail of adenine, while the 5' mean the um, uh, 5 side, that is the prime 5. So this cap and poly A tail is used for the protection from the before the going into the cytoplasm. So the rupturation or damage of the RNA prevention is due to the poly A tail and 5 cap. So that's produce the intron and exon is the black color show is the intron, and while the exon is the white color. So this splicing mechanism to splice and cutting of the intron by the splicing and the ligase enzyme used to bind again this RNA, and that means the produce and poly A tail and 5 cap is the 5' cap is the produced, and that is the exon, and these are the coding segment which that is decode into the cytoplasm and go into the cytoplasm. So the 5 cap, so 5 cap is the major part of the transcriptional movement of the RNA. So that's important. And here is the another thing is the nucleolus is also produce the pre-ribosomal RNA and transfer RNA, which that is go into the cytoplasm and after the modification to produce the ribosome and transfer RNA. That is also produced from the nucleolus. But here is the overview. We will not discuss about that. I hope I make a new video about the translation and transcription to understand well. So here is the RNA will go into the cytoplasm. So the regulation of messenger RNA is the degradation. So the degradation, the messenger RNA lifespan in the cytoplasm is hour to week. So that is the depend on the situation. So next is translational and post-translational control. So that is the next point. After the translational control, what will be the mechanism about that? So here I am writing that translation in eukaryotes cells involve many protein factor like initiation factor. Thus there are opportunities for the control of gene expression at the level of translation. So that's after the level of translation, that is the control by the different mechanism we will discuss in this time. So just remember that is the ligand which that is used for the transcriptional factor, while that is here is the nucleus and here is the cytoplasm. So for example we are taking the steroid hormone or something which that is the primary ligand, and that is here is the hypothalamus and here is the adenohypophyse neurohypophyse, that is the pituitary gland. So that is we are giving the example, we are taking the example for understanding. So just remember about that. So here is the for example corticotropic releasing factor will stimulate from the hypothalamus to pituitary gland that produce the corticotropic releasing hormone, which that stimulate and that stimulate the adrenal gland, and after the stimulation of adrenal gland, that cortisol will be produced. So remember, cortisol hormone is the lipid soluble steroid which that is passed directly into the plasma membrane into the cytoplasm, and when go into the cytoplasm ultimately also go into the nucleus, and when go into the nucleus, that means that steroid lipids soluble direct transport into the nucleus, and that means here is the part steroid receptor in absence of steroid. So in the absence of steroid, this is the inhibitor protein which that bind, and hormone receptor site is here. So when we'll bind this cortisol hormone, for example, or another protein, when bind with the hormone receptor site, so that will be releasing of the inhibitor protein. So that inhibitor protein will be released, so that DNA binding surface will be formed. So this is the DNA binding surface will be formed. So after the DNA binding surface will be formed, so the transcriptional factor will begin. So this is the transcriptional factor modification will be occur. And so this is the steroid or hormone which that is bind and go into the enhancer sequence site of the DNA. When go into the enhancer sequence site to begin the transcription, so this transcription to produce RNA. So that's RNA. I have also told you before. So that is splicing mechanism. So the transcriptional factor attach the RNA polymerase to produce RNA, and that's RNA going in the under the splicing mechanism to produce the 5' site and poly A tail. So that poly means many adenine are present, and 5' site is the basically another part we will discuss in the another video for during the translation and transcription detail. So here this is the messenger RNA will become and modified that's go into the cytoplasm, where the cytoplasm, that is the translation factor, will be bind and begin to translation. So the translational factor is known as the ribosome. That's a two subunit of the ribosome will bind with the RNA, and transfer RNA also bind with the A site, and P site is the peptidase site where the peptide bond will be occur, while the A site is the addition site, and E site is the exert site of the transfer RNA. When exert of the transfer RNA in the E site, messenger RNA will go opposite direction. For example, where the E site the transfer RNA release, so the messenger RNA will if the E site is the left, so the messenger RNA move right. So remember about that, because it's the 3 codon to bind with the anticodon from transfer RNA. So there are many factors will be create, and that is the give the response. For example, hormone, steroids production, secretion, glycogenolysis, synthesis, insulin production, insulin protein, glucagon, growth hormone, lipolysis, development, cell division, cell death, specific response, and stimulate something, suppress something, activate something. So that is